Good afternoon, everyone. My name is Brian Munsky, and I'm an associate professor in chemical and biological engineering and in the School of Biomedical Engineering at the Colorado State University. I am super grateful to Ridro and Sri for putting together this awesome living history session. And I do actually see the irony that this talk is not going to be given by a living person, but at least I can start with a little bit of history. Specifically, I want to talk for a few minutes about the QBio conference and QBio summer school, which started about 18 years ago in 2007. This was a 2-week summer school program at Los Alamos National Laboratory and a 3-day conference that followed immediately at St. John's College in Santa Fe, New Mexico. This brought together some of the world's biggest innovators in applying physics-based methods and engineering approaches and models to biological processes in systems and synthetic biology and trying to understand very complex biological processes. The aims of this program were first to advance predictive modeling of cellular regulatory systems by providing in-depth training in mathematical, statistical, and computational techniques that are important in systems and synthetic biology. And second, and equally important, they sought to advance the careers of researchers who were junior and new to the field of quantitative biology. Now, I was actually a student in this first class in 2007. And in fact, that's where I originally met Sri, who was one of the main organizers of this particular session. John Doyle was also there for the meeting. I'm pretty sure he showed up to my talk wearing bike shorts after having just ridden up to the ski bowl in Santa Fe and back down. Anyway, John also gave the keynote address at that very first QBio Summer conference, and he referred to it as a watershed moment in modern biology, bringing the methods of physics into the study of biological processes. Anyway, I was a summer school student in that very first session. I was a lecturer in the second session, signed up and joined a postdoc with Ilya Nemenman in order to continue participating with QBio. By the third year, I was an organizer. By the fourth year in 2010, I was the main organizer of the QBio summer school. My postdoc advisor had left the lab at that time in 2010, and back then we still lived in a democracy, so I had no one to advise me to focus on research or to avoid pouring my entire heart into this service project that was so squarely focused on diversity, equity, and inclusion. Every year, hundreds of students and faculty applied to attend the QBio Summer School and the QBio Conference. Over time, our campuses were offered not only in Los Alamos, but in Santa Fe, New Mexico, Albuquerque, New Mexico, San Diego, California, Fort Collins, Colorado, Nashville, Tennessee, and Houston, Texas. And year after year, dozens of our current QBio students would converge together at the QBio conference, whether that was still held at Santa Fe or when it moved to Blacksburg, Virginia, Nashville, Tennessee, Brunswick, New Jersey, or Houston, Texas, or even in San Francisco, hosted by Wallace Marshall, who is joining and hosting our session here today, and Fort Collins eventually. Each year, we offered a packed but stimulating program with lectures, software tutorials, career discussion panels, open-ended projects, group outings, picnics, hikes, dances, and late-night coding parties. The strength of QBio has always been its highly welcoming and very diverse community of scientists who are applying quantitative methods to understand biological systems. So many have come back year after year, and I'm so excited to know that the heart of this program and this tradition is continuing in DBIO in APS. The QBio community that has led to lasting relationships, postdoc opportunities, numerous papers, successful grant applications, and at least one marriage that I know of. I wanted to talk briefly about a couple other products that have come out of these meetings. The first thing I'd like to mention is the QBio textbook, which you can see a picture of here. This textbook, edited by Bill Havacek, Lev Simmering, and myself, includes 30 chapters and has contributions from 75 different authors, most of whom are physicists and engineers working in quantitative biology, and most were formerly QBio summer school students and lecturers. Here's a list of some of the authors, and hopefully some of them are in the audience here today. And the second product that I'm excited to announce is a new set of free electronic course materials that we've been preparing for the community to help introduce more junior students into the field at an earlier career stage and to make QBio ideas and approaches more accessible, even beyond the reach of our well-funded research universities. So we focused on building a set of open access electronic materials, including Python notebooks and tutorials, auto-graded coding homework sets, challenging research-like projects, and career discussion panels geared toward helping more junior students succeed in quantitative biology. The first version of these resources is available right now using this QR code right here. But as sad as it is for me to say this right now, I need to step down from running the QBio Summer School. QBio Summer School has been and will always be my most significant contribution to science. But it is time for new ideas and new energy. I'd love to help any one of you in any way that I possibly can to take over and bring your ideas into this program. Please contact me at the address at the bottom of your screen if there's any way that you can help to keep this history alive. I really wish I could be there in person right now in Anaheim, and I really wish I didn't have to be recovering from this surgery. And it really builds my heart and strengthens my heart to know that the original QBio tradition and approaches and the just community that was formed through these programs is still alive and well in the APS division of biological physics. I want to express my eternal love and gratitude to all of you in this community. And in that spirit of love for this community, I would like to serenade you all with a presentation that I gave to our students in the final day of the final QBio Summer School at Fort Collins, Colorado last summer. Good afternoon. Thanks for coming today. Hope you enjoy what I'm about to say. I was tasked to speak on something vital. So this is what I chose for my title. How crappy can our experiments be without sacrificing our certainty? I will thank my close colleagues in due course, but first must acknowledge my funding source. And let me offer sincere apology for the awful ensuing poetry. And there's no need to hate on GPT, because these shitty rhymes are entirely me. To launch my crappy experiment quest, let's first review some studies at their best. Great labs, Stasevich, Werner's, and Newart's include some true microscopy experts. Their single-cell central dogma displays reveal the lives of genes, proteins, and RNAs. Tracking single cells, both human and yeast, gene regulation is a visual feast of signals, transcription, and translation, every step of the cell's regulation. With osmotic shock, red MAPKs fly from the cytoplasm into the nuclei. At their large numbers, kinase noises small. The red signals equal within them all. Here the central limit is formal. Distributions are Gaussian normal. Yet since GFP's on a single gene, STL1's heterogeneity is seen. With SM fish, we can see, find, and count which cells have RNA and at what amount. Live cell tracking for a single gene site brings polymerase action into the light. Before phosphorylation, it is blue. But in seconds, green shows active Pol 2. A minute past the polymerase firsts, red MS2 shows RNA in bursts. Here with transcripts in red and protein in green, even nascent translation can be seen. Their single transcript shows blue and green frames and ribosome frame shifting, it proclaims. To understand these cell variations, it helps to do a few calculations using a simple but powerful tool taught in every year's QBio school. But if you missed it, please do not dismay. Our previous lectures are here to stay. If you follow this QR, you will see Python notebooks and tutorials free to process single-cell microscopy and to build models to match variability. The single cells in the data you saw showed random differences among them all. Models that account for variation better maximize our information. For single-cell response fluctuations, we use chemical master equations. These describe how probability flows, predicting where the distribution goes. While most CME solutions aren't known, a few approximations have been shown. One bounded approach is the FSP, which I derived back in my PhD. The FSP can the CME solve and its sensitivity resolve. Log likelihoods point out the model best, and Bayesian analyses can rank the rest. That's great for fitting single-cell data. Like this example, which I'll explain later. When systems are completely understood, models fully capture their data should. Mechanistic models can't be inferred with accuracy that's almost absurd. ODEs describe kinase ebb and flow, squares the data, and lines the models show. FSP captures transcript statistics, gene action versus drugs, and time it predicts. Bursting models match transcript dynamics, including polymerase mechanics. Our time series fluctuation fitting captures all dynamics it's exhibiting. A simple model for RNAP predicts the amount of labeled CTD, the amount of produced RNA, and for how long the polymerases stay. Cross correlations give information about transcriptional elongation. HACCP models of nascent translation unlock the secrets of regulation. How do drugs cause production to abate? What's the codon-dependent stepping rate? Propose new mechanisms and clear insights for internal ribosomal entry sites, or explain virus use of different frames. These are just a taste of our model's aims. By finding these models, we can foresee biology's heterogeneity. At single cell, our studies always find gene copy noise is never far behind. When models take noise into account, we maximize information amounts. Hypotheses capturing noise in full provide a powerful scientific tool. Well, that's neat. At least I thought it was cool. So I started a lab like some dumb fool. We predicted gene expression's glory, but there's much more to this much longer story. Experiments can be run in myriad ways. Some take a few minutes, others take days. All assays have errors, some large, some small. And obviously, we can't do them all. If data is too noisy, knowledge is lost. Yet, better measurements come at a cost. How good do experiments need to be? Remember, nothing in science is free. Designing which tests to skip, which to use, requires statistics for how we should choose. Next up, focus on a few design goals, but also present some general tools that you smarter folks can later extend to decide what to measure, how, and when. Let's illustrate an example as shown. HeLa cells after dexamethasone. As a synthetic glucocorticoid, dex is a medicine often deployed to reduce excessive inflammation, for which dex is standard medication. When applied, dex is an activator of the glucocorticoid receptor. After adding dex, GR translocates into the nuclei with dose-controlled rates. Once there, GR binds to the GRE, thereby inducing gene activity. GR induces much of the genome, but that needs to wait for another pull. Among those genes, let's focus on just one that's important to inflammation. The dual specificity phosphatase, DUSP1, many a critical role plays. A key gene in medical processes, high DUSP1 inflammation suppresses. We seek a simple model that predicts and quantifies GR and DUSP1 tricks. Must capture GR transport through the cell and predict how DEX affects this signal. Fit DEX, GR, DUSP1 without a seam and predict the DUSP1 response downstream. Must capture variations in space and time, and today it must do so in rhyme. Because today's research funding looks grim, we also need to find places to trim, to reduce experiment complexity without aggravating uncertainty. With immunocytochemistry, Eric found GR's spatial history. When DEX is applied, GR translocates from cytoplasm to nucleus with dose-controlled rates. The high-dose response is quick and complete. At low dose, it's delayed and much less neat. Eric measured GR in thousands of cells, changing DEX and timing in many wells. Fitting these measurements, he discovers data and model closer than lovers. Here's what the model for GR predicts versus time and concentration of DEX. You can see for all time evolutions, models match data's full distributions. Here are means and variances versus time. Once again, we see the models do fine. The squares show the average statistics. Solid lines show what the model predicts. Vertical bars show true deviations, and shading shows our expectations. Since DUS1 understanding's our wish, Eric measured single cells with SM fish. These measurements quantify many insights: cells, nuclei, RNA, transcription sites. Counting many cells using Luis's programs, we computed gene expression histograms. The biological knowledge that Eric sought determine which cells respond and which do not. Here's the match to the RNA DUS1 once a fit to some data has been run. Here the model is orange, data are blue. How much better could we possibly do? Note, these fits are made at 1 DEX value. The rest we predict to see if they're true. DUS1 predictions pretty good for 7 orders of magnitude. Again, these results really do please me, but getting this information wasn't easy. It shames me to say that the level of work that Eric did here would drive me berserk. 12 time points, 9 levels of induction, each 1,000 cells, quite a production. Every cell segmented, imaged, counted, as our time and expense quickly mounted. 10,000 cells, he counted RNAs. It seemed to take years of months, weeks, and days. Our lab's okay, but funding's not vast. Is there a way this could be done faster? Love the results, but would I be happier if we'd reached them with experiments crappier? To answer these questions, we need more math to optimize our scientific path. Through an FSB calculation, we calculated Fisher information. The FIM yields a powerful test to determine which experiment's best. With FIM, it's easily found, the eminent Kramer-Rao lower bound. It defines how good inference can be through asymptotic normality. Each eigenvalue and vector pair shows how well we'll learn parameter combos. Consider experiments you could do and compute FIM for a few. The one with the largest determinant is the assay that's most discriminant. Its MLE estimate will be tight, more likely to get your parameters right. Or if you have some estimation goal, use FIM to find the cheapest tool. Let's return to Eric's GR data. Can models make efficiency greater? Here is Eric's experiment design. For how many cells to count at each time? Sequential FIM analysis found. How many cells to measure in each round? Hey there, scientists, the FIM tells. Use simpler conditions and fewer cells. No prediction accuracy is lost, but it takes far less time, effort, or cost. Predictions for the points that were held out are as good as before, without a doubt. Look at the summary statistics again to see just how well the model does when, using data sets a fraction the size, from experiments easier to realize. Uncertainty metrics, always a must, help us to know which parameters to trust. Bayesian analyses is expected to sort models between kept and rejected. For this, use Markov chain Monte Carlo, Metropolis Hastings, standard, but slow. MCMC samples visually show which combos are vague and which sets we know. For confidence in intervals interior, small would be good, large is inferior. For constrained values, uncertainty is low, as Josh's tight MCMC plots here show. For others, knowledge is a little loose. Please use caution in these parameters use. Whether the intervals are loose or tight, Alex's FIM got the uncertainty right. But where MCMC took hours to run, the FIM in a second was done. Move from GR to DUSP1 RNA. Can we also have improved that assay? Before trying to make experiments worse, let's agree what is a good model first. Our model should fit our data like shown. Tight estimates ought to be known. Once again, FIM matches quite well and has interesting insights to tell. For K-on and GR, numbers are tight, suggesting we get these parameters right. For K-off and KR, they can be debated, but their uncertainties correlated. Transcription rates impossible to find, but burst size B is clearly defined. The FIM works, so let's cut to the chase. Ask how much of Eric's time did I waste? The FIM suggests the best designs should be done better with a fourth of the times. So, would that design actually work? How can I test without being a jerk? Won't make him collect more. It wouldn't be fair. But how can we do with what's already there? Using the FIM, minimal designs. One level of DEX at only 3 times. Small data gives enough information to predict the entire DEX titration. Our work cut by 4, we are fit to burst. But I would like to do a little bit worse. If spots were miscounted when labels fail, would then our modeling be doomed to flail? To visualize labels mistaken, here's an image that Linda had taken. In these images, 2 labels are seen, one in magenta, the other in green. To the same RNA, both should have bound, but not all targets have fluorophores found. Most spots appear in pink and in green. And these ones can reliably be seen. But some appear only with SM fish, while some MS2 spots are clearly missed. We count them all up to see what we find, depending upon how spots are defined. Results are similar, but not the same. How will it affect our inference game? Plotting the green versus red spot amounts, we can calibrate to compare the amounts. From their scatter, define the PDO, the probabilistic ratio. I will refer to this matrix as C and briefly describe its utility. It separates out labeling error, removes bias, and makes our inference fairer. C can be defined for any error, labeling, imaging, etc. Example, if we don't bother to count, you use just the total fluorescence amount. C just quantifies that distortion effects, predicting what insight it hides or wrecks. We add C to the FIM design. Skip much of the math because it's hard to rhyme. The key point is that FIM is linear, making all operations far easier. Now let's get back to our DUSP1 case. How do errors affect our estimates? The distorted inference shown in blue does nearly as well as with the data true. Lost but a small information portion when PDO accounts for the distortion. But what if we made it a whole lot worse? Using imaging that is much more coarse, so that we cannot resolve spots at all and estimate from fluorescence total. For this experiment with way smaller cost, green shows how little uncertainty is lost. Until now, FIM only compares to select between experiment pairs. FIM is even more valuable when used to achieve a specific goal. Next, we asked, How many cells would we need to use this much cheaper microscopy? If we do the crappiest assay times 4, red shows we achieve the required uncertainty score. So that ends my poem. Let me conclude. When we started, our experiments were good. Like Picasso in his earliest phase, we sought to perfect our technical ways. But having verified competence first, our next goal was to make everything worse. FIM analysis quickly decides under which condition the most insight resides. PDOs account for data distortions, retaining the most relevant portions. When integrating models from the start, experiment design becomes a rigorous art. Yet with cubist science, we can be relaxed while solving the model inference task. While I am obsessed with crap, make no mistake, these coworkers of mine are truly great. Now in my poem's penultimate rhyme, let me thank them again one last time. And though I am sad that now I must go, let me cheer once more for DBIO.